Mic check, 1 2 1 2. Mic check, 1 2 1 2.
Hello, guys. Welcome to the 12th Journal Club of Antibodies. Today we have a special guest with us, Dr. Govinda Sharma from British Columbia Cancer Agency. And just a brief intro in case you didn't read our post before, Dr. Govinda Sharma, he's a postdoctoral researcher in Dr. Robert Holt's lab, and he works with high throughput T cell antigen discovery, which is pretty unique work, I'd say. Not a lot of labs that I see who work on T cell epitopes, but at the same time, I can imagine very huge consequences, good future outcomes from this work. Uh, Govinda, you mentioned that you are a major movie buff.
I try.
So does that mean you just watch a lot of movies, or you have very critical opinions on movies?
Yeah, I try to. Well, I mean, both. Obviously they're for fun, but I try to watch with a critical eye. Some of the podcasts I follow are like movie critic
Mm-hmm.
podcasts, so I try to get into that world. I sort of feel a bit of a kinship, with film in that way because a lot of what we do in science is just storytelling. So
Mm-hmm.
there's some tips and tricks that you see in that world that actually what we inadvertently or on purpose apply here. So it's
Yeah,
it's
yeah,
a
right.
and I try to go back and watch classics where I can too, like the ones that people always talk about. There's so many movies that people talk about but haven't seen. That's kind of so I try to make sure I've seen all those.
I watch a lot of late night shows, and I use some of those comedy punchlines in my talks.
Yeah, exactly.
They
There you
don't
go.
work on Zoom. Never try that on Zoom because it's just very awkward. Don't even know if people are laughing or not, and I don't. I just continue speaking. It's very bad on Zoom. Works
Yeah,
great in
you need that
real
instant
presentations.
feedback.
Yeah.
Yeah, exactly.
All right, I'm gonna hand the stage to you, Govinda.
Okay, awesome. Is my screen up?
Yeah, your screen's up and
Oh,
audio, everything's working.
wow. All right. So, well, thanks for that introduction, and thanks to everyone who's viewing on the live stream right now and is going to be watching this in the future. Um, uh, so, as Jetman said, my name is Govinda Sharma. I'm a postdoc at the Michael Smith Genome Sciences Center, which is a department of the BC Cancer Agency. And I'm going to talk to you today about the work that I did through my whole PhD and now into my postdoc years to improve TCR, T cell receptor profiling by applying high throughput T cell antigen discovery methods. So, just by way of introduction, I'm coming to you from Vancouver, BC, pictured here, and we're located at the BC Cancer Research Center here, where I work for Dr. Rob Holt. Um, and in the whole lab, we have quite a few focus areas. There's a number of things on the go there. We range from immunogenomics to synthetic biology, microbial metagenomics, and cancer genomes. But as I said, I focus on T cells and T cell antigens. And this work is motivated by the fact that T cells are a really attractive area of study because it's T cells, unlike antibodies or any other part of the immune system, that are responsible for recognizing intracellular targets. And they do this by recognizing short contiguous peptide fragments that are derived from proteins turned over on the inside of the cell, but then processed and presented at the exterior surface in the context of MHC class 1 proteins. So that's illustrated here in this crystal structure representation. You can see at the bottom half we have the peptide MHC complex. In this case, it's interacting with the TCR alpha beta complex. So historically, this interaction has been a really difficult one to study, and therefore it's been a difficult endeavor to do T cell antigen discovery. And this interaction is a complex one for a number of reasons. Firstly, this peptide epitope that's at the center of it all could be one of an immense number of possible epitopes that could exist. And so the first challenge to any sort of T cell antigen discovery effort is figuring out how you're going to sensibly search this massive peptide epitope space. Secondly, this peptide sits in the context of MHC class 1 proteins, as I mentioned. And these MHC proteins, each of these could be one of thousands of different variants that could exist that are coded by thousands of different MHC alleles or HLA alleles that exist in the human population. So the next challenge in designing any sort of T-cell antigen discovery study is figuring out what the identity of the MHC molecule you're working in will be. And then on top of that, this whole interaction, this TCR peptide MHC interaction, is a very low affinity and transient and cross-reactive one. And I'm going to talk a little bit more about cross-reactivity in a minute. And all of these things, these affinities, this kinetic on-off rates, conformational changes in the T-cell receptor, the presence or absence of co-receptors, all of these things come together and integrate into a signal that determines whether a T-cell will respond to an antigen or not. And this is an important thing because very often affinity is simply used as the sort of proxy for immunogenicity, which isn't, as I'll show you, necessarily a fair assumption. So just for that last point, this is a specific example. So these are two different agonistic epitopes, peptide epitopes for the mouse model TCR 2C. It's called 2C. And you can see that when they're overlaid, they have a they're structurally very similar. And indeed, when in complex with their appropriate MHC, they have a very similar affinity for the 2C TCR, certainly on the same order of magnitude. But if you look at their ability to elicit response from the 2C TCR, they actually differ by several orders of magnitude. One is much more potent than the other. So this just goes to show that affinity is not the whole story. Functional activation, and therefore functional testing, is key. So in the face of all of this complexity, how successful have we as a community been at doing T-cell antigen discovery so far? The short answer is not very good, and I'll show you what I mean. So this is the front page of the Immune Epitope database, which is a major public repository that contains the majority of T-cell epitopes that have ever been discovered and documented in the literature. And if you go there and you run a search for T-cell assays of any MHC restriction in human, you see that you get about 90,000, just over 90,000 unique hits. And while that seems like a lot, to put it in perspective, if we look at the other side of the synapse, if we look at what we know about T-cell receptors themselves, we see that our knowledge of T-cell antigens is completely outpaced. So this is the iReceptor database, which is one of a few different databases that contains TCR and actually antibody repertoire sequencing studies. And this one just actually contains a fraction of all the TCR-seq studies that have ever been done. But if you go there, you see that there's over 670 million TCR sequences deposited in that. So we're very good at IDing T-cell receptors by sequence, but we're really not very good at figuring out what they recognize. So this is an unmet need. There's obviously a bit of a gap in knowledge here. And what accounts for that is that the existing methods that we use to do functional T-cell epitope testing are very limited in scope. So what do I mean by that? If we have a look at some of these methods, most of these classic cell-based functional testing approaches require you to either develop a readout from the T-cell itself. So this is the case in cytokine capture methods like ELISpots, or they require you to monitor some sort of activation marker. This could be the case in NFAT-linked reporter systems or other sort of natural activation markers that come up. Or alternatively, they require you to monitor the complete destruction and lysis of your antigen-presenting cells. And that's the case of chromium release assays and the like. So you can imagine if you're in a situation where you have a T cell clone or clonotype of interest, and you would like to test it against a large panel of potential epitopes, these configurations all require you to test them individually, one at a time, in separate wells of a plate, for example. And that becomes very difficult to scale to large numbers of peptides, like in the millions, for example. So by analogy, these methods are a lot like using a telescope to look at the stars. Your field of view is very limited, and finding anything interesting is totally dependent on which part of the sky you're pointed at. What we really need is an unbiased view of the whole sky. And this is important because of, as I mentioned before, important antigens will exist outside of your telescope view because of TCR cross-reactivity. From a therapeutics development standpoint, this is particularly relevant because sometimes these cross-reactivities can be dangerous autoimmune reactivities, or they could be really useful ones that we're just otherwise leaving undiscovered because we have a very narrow view. So just before I go on, let's talk quickly about cross-reactivity. So it's been an open question for quite some time. How cross-reactive are TCRs really? Like there are specific examples and reports of T cells that recognize more than one peptide, certainly. But how prevalent of a phenomenon is this? And there's been a number of studies over the past decade or so that have sought to answer this question, notably this one from 2012, where the authors here just successively subsampled random peptide space and were able to come to an estimate that a single T cell receptor can recognize more than 1 million distinct peptides from random space. Moreover, if you look at these peptides, and this is a piece of a figure from that publication, you can see that these epitopes don't need to have significant sequence similarity to each other to elicit response from the same TCR. All right, so this is an ELISA readout where increasing concentrations of these 6 different peptides were added to the same T cell clone. I can't remember the name of the clone off the top of my head. And MIP1B release was detected as a cytokine release readout. And all 6 were able to elicit response from that T cell clone, despite really not looking very similar. And so the key takeaway here is that every TCR, we shouldn't think about them having an antigen that they recognize, but rather we should think about every TCR having its own repertoire of peptide epitopes that it can respond to. And if you think about it that way, you can see that there was and is a need to be able to do high throughput functional T cell epitope discovery.
So that would be very similar to what we see in antibodies, that they don't react to just one thing, but they do want to react to multiple things. Okay, and
yeah.
Mm-hmm. And
sure. I mean, a key thing here, though, too, is that TCRs don't do affinity maturation.
Right.
So
yeah,
they're always active. They don't hone in on a single target. They remain cross-reactive, and they remain poised to recognize this repertoire of targets that they have.
And I would think that makes sense from an evolutionary perspective, because a cross-reactive TCR would provide a broader protection to the organism than something too specific that it can't see anything else.
Exactly. It's this question of cross-reactivity. It sort of came from the logic that there are only so many cells you can have in your body, and if it was true that every T cell could only recognize one antigen, then the number of T cells that you would need to provide an adequate protection against the whole universe of possible threats that are out there would require the organism to be just unwieltably, untractably large. So cross-reactivity must exist. And so, characterizing it has been the challenge in recent years. So that brings us to the publication of the day here. So this was our Nature Communications paper that came out in October of 2019. This paper had a long road. So some of the data I'll show you was generated as early as 2014, and the last pieces that went in, were generated in 2019. The very first version of this manuscript that we ever submitted, we submitted in the spring of 2017. And after almost 1,000 days and getting bounced between 5 different journals, we finally found a home for it at Nature Communications. I can't tell you why this was such a tortured process. I think it's just my luck, but very happy with how it turned out. I think it's a great paper, and let's get into it. So I'm going to use the term tope-seq. So this was always like an internal term that we use. It's short for T-cell epitope sequencing, and this is the name of the method that we describe in this paper. So this is a story about the development of a new method to do high throughput function-based T-cell antigen discovery, and we were successful in developing a method that we call tope-seq. And it works by leveraging the exquisite specificity of the granzyme perforin pathway. So in vivo, when a T cell recognizes its target cell, it's able to deliver a class of proteases called granzymes, and predominantly granzyme B, to the intended target cell very specifically without allowing any to splash into nearby healthy bystanders. And so we took advantage of this property by building a fusion protein that consists of cyan fluorescent protein and a yellow fluorescent protein fused together but separated by a granzyme B cleavable peptide substrate. And so then this whole transgene could be encoded, or was encoded, in tandem with short peptide coding minigene sequences. And the whole cassette was delivered as a lentiviral vector to host surrogate target cells. And so once inside those target cells, each of these components would be independently expressed. And if we interrogated target cells that were expressing this intact reporter protein with a violet laser, what we observed is that you get, instead of getting totally just CFP light coming off of this cyan protein, some of that energy would be transferred to the yellow moiety of the molecule, and we would observe this as a yellow color coming off the violet laser. And so this is a phenomenon called FRET. It's widely used in biology and other things. And we can monitor this signal in FACS and flow cytometry by plotting this FRET emission signal versus CFP. And you see you get this resting FRET signature in untouched target cells. So if there's a minimal epitope present in our minigene sequence, it can get processed and presented and potentially recognized by the T cell of interest that we're co-culturing with, at which point granzyme B gets shuttled over to the target cell specifically. A reporter protein is cleaved. And again, when you look at cells with cleaved reporter under violet laser, you see that you rescue that free CFP emission and you lose FRET transfer. And this too can be monitored in flow cytometry, and it manifests as this very pronounced FRET shift that allows us to readily gate on cells that are actively being targeted by T cells. So then this fits into the idea of doing high throughput library screening, because then we can build large libraries of mini genes into lentiviral vectors, deliver them to our target cell lines such that each individual cell has a different mini gene inside of it, and co-culture it against whatever T cell of interest you have, performing the FRET shift assay as I just described, and actually isolating putatively antigenic target cells, amplifying by PCR the mini genes that are encoded within them, and then using Illumina sequencing to ID the mini genes that were responsible for eliciting T cell response.
I have a question at step number 3. So are you looking at almost dying cells that have just received granzyme?
Yeah, so these are cells that have received granzyme, and as I'll show you in a couple slides, they are very early apoptosis, so early, in fact, that we can sort and manipulate and basically work with these cells before they sort of fall apart and die.
Okay. Do we know what's the time window between granzyme injection and complete death that's fragmented?
Yeah, I'll show you that
Okay.
in a couple of slides here. But before we get to that, the first proof of principle that we did to actually, show that this works and to show that granzyme specificity that we're after is preserved in our in vitro system. I didn't go straight into library screening approaches, but rather I used this binary mini gene approach, and I used the model mouse system OVA, OT1. So OVA is chicken ovalbumin protein, which is known to be presented on mouse MHC cells or mouse MHC molecules and recognized by the model mouse TCR OT1. So I built a mini gene that contains the minimal OVA epitope known to elicit response plus some flanking sequence. So this wasn't the whole OVA protein, it was a mini gene. And I built a scrambled negative control in parallel, which is the same OVA fragment, but just with the minimal epitope scrambled. And I used each of these mini genes to build viral vectors that I could then transduce mouse host cell line with to create OVA mini gene expressors and scrambled control cell line. Mix these cells together at a 1-to-1 ratio, and then co-culture that mixed population against cytotoxic T cells from the OT1 mouse, OT1 TCR transgenic mouse. And the results of that are shown here. So down the middle is the Fret shift assay of this co-culture. And I used FACS to isolate both this shifted targeted population and also the untouched population, the population of target cells that was not exposed to T cells at all. And in either case, I assessed them with qPCR using a custom TACMAN assay that could distinguish between the scrambled control or the OVA mini gene. And we can see that in the untouched control, all the mini genes that are detected, both the OVA and scrambled come out at about a 1-to-1 ratio as expected. But in the targeted gate, over 95% of the mini genes that were detected in that gate are the correct OVA sequence. So this indicates to us that we were in fact very specifically able to detect the right sequence. And so the next question that we wanted to address was the same question you asked, Chetan, is how do we know that we're not losing significant numbers of target cells due to apoptosis? And so to address that, I did a simple time course experiment, using the OT1 T cell and OVA mini gene expressing targets. And I set up co-cultures for a variety of time points and at a variety of different effector to target ratios. And I monitored two things. I monitored the Fret shift signal relative to background, and also measured the PI signal. So PI or propydium iodide is a viability dye that's commonly used in flow cytometry, and it's a DNA binding dye that cannot penetrate the cell membranes of living cells. It only becomes permeable to cells that are undergoing apoptosis and therefore have compromised cell membranes. So in other words, PI positive cells are cells that are in apoptosis. And we can see that if you follow this time course along, there really isn't any PI signal until about 12 hours, where it very abruptly spikes and then comes back down as these cells start to actually fall apart and drop out of culture. But in parallel, you can see that the peak Fret shift signals that we got preceded this PI spike by several hours. You can see that the peak tended to come at around 6 hours. And actually, just to be safe, for all subsequent experiments, I used a 4-hour time point at a 1-to-1 ratio. So that we had very, very good confidence that we weren't going to be losing significant target cells in any library screening experiments.
Okay, so I have a question now. So we know that PI binds within the cells which have a fragmented cell membrane, but before that, I don't know how much time before that the nucleus started to condensate. How can we establish when the nucleus was started to fragmentate and etc. or something like this? Is a while before the membrane starts to losing its integration, right?
Yeah, well, so our objective here was to be able to sort. That was the key manipulation that we really needed to be able to do. And so, you know, PI cells it's kind of it expands a lot of the apoptosis cycle. And so we know we can actually see PI positive cells. They're still intact enough to see in flow cytometry. And so anything that precedes PI loading, you know, we conclude that allows us to actually sort and recover these cells. But then the next step after that is actually to lyse them and recover their genomic DNA and PCR out the mini genes. So we're not actually all that interested in the long-term health of the cells. We're not going to culture them per se afterward. I mean, you could try to. That would be a really interesting thing to try to do. But the goal here was to have them intact enough and healthy enough to actually recover them, be able to recover genomic DNA, and then isolate these very small mini gene sequences that are integrated into that genomic DNA so that we can proceed into sequencing. So we weren't really too worried about the status of the nuclear membrane or even the status of the genomic DNA itself, as long as we could recover and ID the mini genes. So
I have
so
another question.
Sure.
Sorry. Did you ever try treating the cells with an apoptosis inhibitor to kind of elongate that window, like ZVAD or something?
Yeah, that's a great question. Yes, I've tried a number of them. The problem is that a lot of these caspase inhibitors of all sorts, whether they're peptide-based or coumarin-based or what have you, also inhibit granzyme B to some extent. Mm-hmm. So you kind of kill the signal, but you do elongate. So you can imagine there might be a sweet spot in there, or maybe a way to actually integrate those inhibitors into this process. And I've done some work on that, and I still think that there would be a way to do that, but so far no, because of that issue.
Right. Okay.
So at this stage is when I went into testing this approach out in a library-based context, and I did that by generating random mini-gene libraries, starting from totally degenerate oligo sequence, so just a string of ends, and then cloning that into a lentiviral transfer vector. And I generated from this a library of more than just over 1 million unique mini-gene sequences that I then transduced into the same host mouse cell line as used previously, again, such that each individual cell in this library population expresses a different mini-gene. And then into this random mini-gene expressing line, I spiked in the OVA expressing cells from the previous experiment at decreasing abundances, starting from 1 in 10 all the way down to 1 in 100,000. And then each of those spiked populations I co-cultured with OT1 T cells. So the results of that are shown here. So this time I collected, again, the shifted gate, the targeted cells, and also the unshifted gates, the cells that were not undergoing fret shift readout. And instead of qPCR, I subjected these cells to standard PCR and then Illumina sequencing. And the results of that are summarized here. So if we start at the left side of the screen, we can see that at the highest abundance in the 1 in 10 ova spiking condition, if you look at the mini genes that were detected in the shifted gate, the majority of them were ova mini genes, and that ova was substantially enriched in the shifted gate relative to its counterpart unshifted gate. And this is a trend that continues all the way down to the 1 in 10,000 condition, where if we zoom in, and that's what's shown on the right side here, if we zoom into 1 in 10,000, we can look at all the mini genes that were detected. So here, each dot is a unique sequence. And if you look at the relative abundance of each of these sequences in the shifted gate, minus its relative abundance in the matched unshifted gate, you see that most of the sequences fall along the 0 line because most are not enriched due to T cell pressure. However, the ova mini gene is significantly enriched due to T cell pressure, more than 10 standard deviations above background. So this result indicated to us that the TOPE-seq approach has a sensitivity that matches the conventional gold standard functional testing methods, but with the added advantage of being able to screen more than 1 million different unique sequences in parallel.
Can I ask something here?
Yeah.
So there are some few more dots that are on the same y-axis plane as the OT OVA. So does that mean OT1 might be cross-reacting with those?
That's what we hoped. So we actually went and looked at these other ones that came up, and it turned out in these cases that those were spurious hits. They didn't actually elicit any real response. So there is a bit of background that does come through, but one thing we actually did in subsequent experiments that are actually described in the paper, but I won't spend too much time on here, was that you can basically shave off the top however many you want mini genes and have them resynthesized on an array-based platform, and then rescreen those, and that's sort of analogous to panning, that you would do in the antibody world. So you can do a second round panning screen. And when we did that in different experiment, we found that even just on the second round, the signal was almost entirely clean, or it was entirely cleaned up. So applying panning to this really improves the signal to noise that we see. But even on a first run, without any panning, this was a pretty clear indication that we could pull our test antigen back out of a library background. So the next question that we were often asked, presenting this work outside of our lab, people would often ask, okay, well, that's great if you have a monoclonal T cell population ready to go as an input for any sort of screen, but that's a really difficult process to get to a clonal population. How well does this approach stand up to polyclonal T cells, to a mixed population of different T cell clonalities? And so we address that by taking a similar approach and doing a spike-in series on the T cell side. So in this case, I spiked in OT1 T cells into a background of just wild type Black 6 mouse T cells. And in this case, I used 1 in 30 and 1 in 3,000. And then co-cultured those mixed populations with the spiked ova minigene random minigene libraries at either 1 in 100 or 1 in 10,000. And I also actually repeated this design in a second model mouse TCR, the PMEL1 TCR, which is an established melanoma reactive T cell in mouse, and it's known to react to the human GP100 protein. So I built out these materials and did this experiment with those cells as well. And we found that in this sort of the easy mode, we call it, the 1 in 30 and plus 1 in 100, it was slam dunk. We were able to very easily pull out the test antigen from background. But even at 1 in 10,000 library abundance, the 1 in 30 spiked T cell population was very readily able to detect either of these test antigens above background. almost as well as the monoclonal population, actually. So this actually was a very strong indicator that not only is the tope-seq approach sensitive, but it's also robust enough to polyclonal input population that it could be really practical as well. And just to extend that further, this question of how practical and useful is this approach, we extended this a step further. We said, okay, we know we can screen polyclonal populations, but can we screen polyclonal populations from an actual real biological context? And so to test that out, we grafted a set of mice with of just wild type immunocompetent black 6 mice with the B16F10 tumor model. And these B16F10 cells that we used, we had actually transduced with the OVA protein as well. So we knew OVA was in there. We engrafted those tumors and we let them grow out for about 3 weeks, at which point we harvested the tumors back out, dissociated the tumor cells, and isolated the tumor infiltrating T cells. So the ones that actually were getting in there and responding to the tumor. We expanded those out ex vivo and then screened those directly against the 1 in 10,000 OVA minigene spiked random minigene library. And when we did that, we found that in all 3 of the mice that we tested, again, we were readily able to detect OVA as highly enriched above background by tope-seq. So these data then indicated to us that we could take polyclonal T cell mixture from a probably suboptimal biological context. We know the tumor microenvironment is not a great environment for T cells generally, and still find a tumor rejection antigen by this method. So this is this was a really exciting result, and this is where we conclude the paper. This is sort of where the publication ends. But we've still been working on this, and actually in 2020, so as I said, this paper came out in October of 2019, and we got some good news on this work in 2020. In April of 2020, we received the full issued US patent on this tope-seq methodology. And then a few months later, in December of 2020, we received the Canada patent. And both of these patents have priority back to 2014, which is when we filed the first provisional filing on this technology. So even that was a 6-year process from first filing to actually having something fully patented, which is not unusual, but that was also another sort of long story that was happening in parallel to this as well. So I did bring some slides of what I've been up to since the paper. I don't know if we want to get into that or if we want to stick to strictly journal.
Oh no,
club.
yeah, sure. I'd be down for this.
Sure. Okay. So I mean, obviously
I was a member of a synthetic biology students club for like 3 years, so maybe my friends will be happy to hear about it too.
Great. Hopefully I can give them something interesting to ponder here. So
I have some one specific question about what we just talked about before.
Sure.
How do you get one cell to have only one minigene? Is it expected of them to have one minigene, or are you like, what's the method behind it?
Yeah. So that's a good question, and that's actually something I spent quite a bit of time on. So it's just straight up Poisson distributions. It's Poisson statistics. So a lentivirus you can have more than one lentivirus particle infect a target and integrate. So you can have multiple copies. And so I developed a method of titering any lentivirus that I produced, so I could have a very like a characterized number of transducing units per unit volume. And I basically used a very low MOI. I generally aim for an MOI of about 0.2. which means that most of your cells that you apply the virus to will just be empty. They won't receive any transducing units. But of the ones that do, you would expect probabilistically that more than 95% of them would be singletons.
Okay.
And then, this was a thing I also confirmed because I developed a qPCR assay that I could use after transduction and purity sorting to confirm that the copy number of mini genes I was putting into my library cells on a per cell basis was around 1.
Okay, so it's mostly probability based, and you're going to maximize one copy per gene, but there'll probably be very few cells that are expressing multiple. Okay.
Yeah, it's a lot. It's like a lot of the same types of statistics that you see in things like droplet digital PCR or like in a lot of single cell techniques where you're doing oil and water droplets and encapsulating a cell.
Yeah.
It's exactly the same type of statistics that's done as that.
Okay.
So everything we did in that paper, that Nature paper, was in a mouse model system. I'm sure you noticed. So the next question here was to adapt this whole approach to human cells. And as you can imagine, this comes with some unique challenges in the human system. Namely, how do you get large enough numbers of appropriately MHC matched target cells? And also, how do you get large enough numbers of clonal or at least highly enriched T cells to use as effectors in any sort of high throughput library screening experiment? And so I sought to address these challenges by using a totally synthetic biology approach. And what I mean by that is I took advantage of three properties of cell lines that we had at our disposal. So for one, YTND is an NK-like cell line, an immortalized NK-like cell line that still maintains its granzyme perforin cytotoxicity. So we could use that granzyme as a potential readout. K562 is an MHC deficient cell line, but it still has intact antigen processing and presentation machinery. And in fact, it's actually pretty commonly used as an artificial APC cell line for that reason. And in the absence of any modification, YTNDs don't kill K562s. They just leave each other alone. So the hypothesis here, the question I wanted to ask was, will adding the TCR peptide MHC components to these cells redirect that YTND cytotoxicity towards the antigens or the targets of TCR clonotypes of interest? And depicted visually here, what in other words that means is, if I was to add the alpha beta TCR chains of interest along with all of the CDR or CD3 and CD8 domains that form this TCR complex, would that be sufficient to hook into the signaling machinery of the YTND cell line and produce a granzyme response to HLA and mini genes and mini gene libraries that are transduced into K562 artificial APCs? So that was a question, and I tested this out actually using a whole variety of different model test TCR antigen pairs. But one particular interesting one that I'll show you right now comes from this 2013 study, which you may have heard of. This was big news in the cell therapy world in circa 2013. And what happened here was these investigators took a well-known T cell receptor that responded to melanoma cells. It's called A3. And it was known to respond to melanoma via this MAGE A3 antigen, specifically via this peptide sequence. And so these investigators introduced mutations into the wild type A3 TCR to increase the affinity of that TCR to the MAGE A3 antigen. And this resulted in a TCR called A3A, they called it. And so they use this TCR then as a therapeutic. They took this T cell receptor sequence and by lentivirus transduced it into autologous T cells from 2 different patients, grew those T cells out to very large numbers, and then reinfused them into the patients. So this is analogous to CAR T therapy, for example, but they use this engineered TCR instead of a CAR, with the intention of targeting this MAGE-A3 mini gene and clearing the melanoma. What happened instead was on infusion, both of these patients sadly were subject to very sudden and fatal adverse events. So both patients died at post-infusion. And after a lot of investigation, these authors did a lot of work to conclude that the reason for this was that they had inadvertently introduced a cross-reactivity to a protein called titin, which is expressed in the heart, the living heart. And with the tools they had available to them at the time, they had no way to see this coming. So it was really quite a stunning story. And what I've done here now in 2020 is taken these materials, had them synthesized, and I've built them into the respective reconstituted CTL or K562 artificial APC systems to test that if I co-culture these all with each other, can I recapitulate this TCR reactivity in the fret shift system? And the results of that are shown here. Along the top, you can see that these are each of the MAGE-A3 or the titin cell lines exhibiting their just resting fret signature in the absence of any effector cells. But you can see once you start to add wild type A3 RCTLs or the A3A effectors, we do start to see populations of cells undergoing fret shift. And if you overlay the numbers on top of that, we see that this largely fits the narrative that was observed in 2013. The wild type A3 TCR responds to MAGE-A3, but only very weakly to the titin mini gene, if at all. And the A3A responds much more strongly to MAGE-A3 as intended than the wild type, but also responds to the titin mini gene quite strongly, which was obviously not intended. So this is a positive confirmation of this sort of synthetic cell chassis that we can then now use to plug any TCR of interest that we want into a TOPE-seq approach. And that's actually one thing we're doing right now, is asking the question, can we put this all together? Can we take our cellular reagents that we developed here and the TOPE-seq methodology and develop a self-antigen library and do a screen that would have been able to predict this cross-reactivity long before the clinical trials ever started, and therefore have prevented this catastrophic adverse event. And so that's where we're at right now. Unfortunately, I have to leave that on a bit of a teaser, but this work is ongoing, and I don't have any data to show you just yet. But suffice it to say, this is something we're still very actively working on and developing, and we're keeping this, we're building up this technology platform further since the publication in 2019. So I'll just take a second here to just throw out my acknowledgement slide. These are all my lab mates at BC, at the Cancer Research Center. We have close collaborators in Victoria, BC, just across the water, who we work closely with a lot, and we share a lot of ideas and materials. And then also the various home institutes and funding agencies that supported this work. Sorry, I interrupted somebody with that.
Yeah, I had a question about the Titan, but this was a great talk, and I'm really thought that the new data was very interesting. But the Titan antigens, I'm guessing they're auto antigens, correct? Self antigens.
Yeah. So Titan is that's just the native sequence. Titin is a protein that's expressed in beating cardiomyocytes, so everybody expresses that. There was no mutation or polymorphism. Like, this is just the one that everybody has. The MAGE-A3 as well is a self-antigen. It's a cancer-associated self-antigen, cancer testis antigen. So
Yes.
so these were not.
I would kind of, envision having sort of like a mini gene library of just human self antigens in this context, right? Like, that's how you would screen whether these genetically modified TCR sequences would be able to recognize these autoantigens.
Yeah,
right?
and that's exactly what we've done. So I had a slide in here that I took out right before we went live, but we have built a library at home of that, starting from just normal human genomic DNA from a pool of normal healthy donors, isolating that genomic DNA, shearing it up, and then performing an exome capture on it. So totally analogous to what you would do if you were going to do whole exome sequencing. But instead of putting it on an Illumina flow cell, I cloned it into my lentiviral vector and put that into K562 artificial APC cells, and actually came out with a whole exome expressing cell library with an estimated diversity of about 25 million different unique sequences, which you may appreciate is quite a significant step up from the random mini gene libraries that were done for the Nature paper. So so I actually built that and have now run the fret shift assay on it so far, and actually even sequenced it. We were pretty far down the road with this, but as you imagine, that generates like a ton of data, and we're kind of trawling through it right now and processing it. And we've actually had to sort of develop new software to do this because it's just such a unique data set. But yeah, no, that's exactly right.
And also, coming a little bit more from like a cancer point of view, this is sort of a very elegant artificial system to be able to assess a variety of different human, HLA types, allotypes. But in the case of, let's say, a cancer patient comes to you, and doctors, oncologists, whoever, think that they could be a good candidate for immunotherapy, right? So I kind of see the future of this personalized immunotherapy being that you're screening antigens from these patients, but you're screening them with their own T cells. So do you think this technology, I'm assuming that, this is a little bit less sensitive than what it would need to be to do that. But how do you sort of envision that happening? How do we get there to that point?
Yeah, that's an excellent question. So some of the other things that we've got going on involve taking smaller libraries, libraries of candidate neoantigens, and testing them against pools of T cells that we get from patient or donor blood, with that exact intention of seeing if we can isolate a T cell clone or even just a TCR, that you can then use to produce a TCR T product as a specific therapeutic. We've kind of focused at the moment on looking at recurrent mutations, shared antigens that would be, across a larger population. And this is, I think, like what you're getting at, largely because that it's doing this sort of approach is right now logistically pretty challenging, and it takes a long time. It's not really like fast enough right now to be able to turn around a totally personalized cell therapy product for whoever comes in the door. But the same principle could absolutely apply, right? If you take some sort of neoantigen panel that's specific to that patient, or at least somewhat specific to that patient, and then screen through their own repertoire to find some T cells. That's a possibility. I think like much like antibodies, the beauty of T cell receptors and by extension TCR-T therapy is you don't necessarily need to screen through that patient's repertoire to find the T cell that will work, right? You know, if you can even just use healthy donors, as I said off the top, everyone's poised to respond to a variety of things, right? So in my body, I would speculate that I might have T cells that can respond to KRAS G12D mutants. You know, maybe I've had cells in my body that are KRAS G12 mutants that have gotten cleared out by my immune system. And so in theory, you could absolutely do that, develop these personalized neoantigen products sort of separately and in advance. And as you sort of start to accumulate products, you kind of take the right ones off the shelf. So that, I mean, that's kind of a rambling answer, but I think at the end of the day, what you can always bet on is that, you know, the technology will get faster and better. I mean, you know, not that long ago, the idea of sequencing a genome in 48 hours was like inconceivable. So I think we could get there, that, you know, get to the point where someone's tumor is profiled, we know what kind of mutations are present, and we can have a cell therapy ready for them in some fixed period of time. But right now, what we're in our lab kind of focusing on is these recurrent ones, the ones that we would take a longer time to develop and everything, but then would hit a larger chunk of the patient population potentially.
Right. And just one other question, you know, speaking a little bit more about, you know, having a more like a generalized or across, like the cross-reactive antigens. When we're going back to your mouse data, you probed for the Steinfeckel epitope, but that is the immunodominant epitope. So in your sequencing results, did you find any other, did you find that the other less immunodominant epitopes were also hits, or did you even probe for that?
Well, yeah, no, I mean, we hoped we would find that. We looked for that. Ultimately, what we concluded is that a purely random mini-gene library is too big. And I think what was happening was that we didn't have enough diversity, like a million sequences from purely random sequence wasn't really enough of a subsample for there to be likely very many of the Steinfeckel mimotopes. So I mean, yes, that would be something I would hypothesize, but I think you'd need to actually have a more honed library to do that. And it could still be very unbiased, but it would have to be just something a little bit more biased than pure randomness, which is something we learned in the process of doing these experiments.
Okay.
I have several things to ask, but before that, anybody else who has?
I'm about to get grilled.
Oh no! I really liked. I actually am glad that you went for the Titan study. I think it's like it would be a good story to tell people when you talk to them about how TCR cross reactivity is important. I'm gonna tell that to my friends in class and lose some friends after that, but it's good. I have this question about. Yeah, I have this question about the MHCs. How do you select which HLA you want to put on your this cell line? Because that's a huge variety you can get.
Yeah. So I mean, there's a lot of criteria I guess you could use to select. I think that's one of the strengths of our system. Actually, it's a major strength of this system is that right now, if you're to like make peptides and then pulse cells of peptide and then use them in your ELISpot or whatever, each time you switch to another HLA to study, you have to make those peptides all over again, right? You have to do the peptide prediction, you have to make those peptides. But We don't do that. We just make one mini gene library, and we have a set. Like right now at the lab, I think we have about 20 different K562 lines with different HLAs in them. And you can just take that library and you just pop it into whichever one you're interested in without resynthesizing or retooling really in any way. So that's an advantage of our system. But to circle back to the original question, how do you choose which HLA to work on? I think quite honestly, what we're using as our main criteria right now is like which ones are the most common alleles, which kind of sucks if you're someone that has like all rare alleles. But as I alluded to before, as we get better at this and as we do more and more screening, we'll be able to more rapidly address just about any allele because we'll have our cell bank of K562s that have all sorts of alleles in them with libraries in them too. So for that reason, a lot of the time we're focused on HLA AO201. Okay. That's a common one. This Titan story, it was known that it all happened on AO101 alleles. There's, I think, some literature out there that characterizes the HLA gene pool. Says that there's maybe on the order of like 50 or so different alleles accounts for like a huge percentage of the human population. So we kind of have a set of like the first 50 that we'd want to kind of have as at the ready. But again, it's very customizable, personalizable. If you have a specific patient or patient population that you want to look at, maybe in a certain part of the world, or a certain allele might be more prevalent, and you say, hey, I want to go target this cancer type in this part of the world, you might select an allele based on that specification alone. So I think the key thing is providing a system that's flexible, and then
no, I really like this kind of Frankenstein system where you're putting your own TCRs, your own MHCs. That's how every Resident Evil movie starts. But I like this approach. I had one. I was very curious about one of the initial slides that he showed, that there was these two peptides, almost similar affinities, but one was a super antigen, other one was not. I'm just curious. Do we know why one is and the other isn't?
Off the top of my head, I don't. I would imagine it might be known. It probably is known. I mean, that's a 20-plus-year-old study, and that model is even older.
Okay.
So yeah, I mean, I would expect we probably know, but if I was to kind of speculate, like I was getting at, there are a lot of different parameters that influence whether or not a T cell receptor will recognize a certain peptide, and also what makes a peptide immunodominant. What actually influences whether basically a peptide-coding sequence actually gets onto an MHC protein, and things like expression level is part of it, but there are other things too that we're still even kind of understanding right now, and that's kind of all to say that a really holistic, functional view is still super important, because a lot of these things are just too hard to predict. Cross-reactivity, we can't predict. Like I showed those 6 peptides that don't look anything like each other. Computational tools right now can't predict cross-reactivity. You still actually need to be able to functionally test it.
If there are two things,
sorry, I'm sorry. Of those peptides that you showed that are cross-reactive, I was really surprised because I thought most of the time these cross-reactive epitopes have the anchoring regions that are similar. Like in this example, the Titan and MAGE3, the anchoring region, the anchoring amino acid is the same. But in the other one, it was in the first slide that you showed, they were completely different. So that's what I thought was very surprising.
Yeah, and that's a thing that's being observed more and more often. Is is that you get these kind of clusters of recognition, right? If you're imagining peptide space, you'll have like maybe one set of peptides that have similar anchors or 2 or 3 common positions, and then you'll have another entire cluster that has different anchor positions, different sort of baseline characteristics, but either can be recognized by the same TCR. And there's some X-ray work and SPR work that shows that a lot of this is because when a TCR and a peptide MHC come together, they can bind at different angles, different orientations, and this influences which residues in the peptide and the MHC are the important ones for influencing response in those registers. They call it registers, right? So it's a super complicated thing. There's a ton of biophysics involved. Even the signaling pathways downstream, like what's happening, like how the TCR itself is getting triggered in response to these really low affinity interactions is a really active but really fascinating area. The T cell receptors is just so interesting, so much more interesting than an antibody.
Well, B cell people won't agree to that, but I agree. I have to give you guys credit for this, for TCR signaling and MHC interactions, for things to be so complicated. You guys were able to come up with something that actually works. I think it's a big deal in itself.
Well, thank you. Yeah,
there's so many
we're
parameters.
Yeah, it's especially the YT part. I mean, that was kind of I don't know if this is going to work, but let's try it. And then it did, yeah. which was awesome. And as I said, I've used a lot of different TCR antigen pairs to validate that synthetic platform. It works.
Okay, so do you guys have any other questions? All right, I think this would be a great point to end our discussion. Thanks a lot, Govinda, for talking to us about all of these things. There are at least 2 or 3 core concepts that I've updated today that I'll take with for the rest of my life with me.
Awesome. I'm glad to hear that.
Yeah, I'm going to switch the screen. Everybody watching, guys, thanks a lot for coming. The videos and the uploads will be available on Facebook and YouTube for watching later. See you all later. Bye-bye.